Hello everyone. Today we are going to see genetic organization of eukaryotes. We have seen in previous videos the genetic organization of prokaryotes. The prokaryotes are those in which nucleus are not developed, but the eukaryotes are those cells which are having completely developed nucleus and all the cell organelles. They are very well developed and functioning than prokaryotes. In prokaryotes, we don't have seen such kind of cell organelles. In prokaryotes, there are only the cell and genetic material which is placed in the cytoplasm, which is floating in the cytoplasm. They are having some extra DNAs, that is plasmids, but here in eukaryotes, the cell is completely developed, which is having cell wall, cytoplasm, and in that cytoplasm there is a nucleus which is well developed, and all the genetic material is placed in the nucleus. So here is the genetic material of eukaryotes. The basic fragment or basic unit of this genetic material, that is DNA. The DNA is tightly coiled and it contains chromosome. The chromosome, which is having three important unit or three parts of the chromosome, that is centromer, then arm, and telomere. So with this supercoiling in eukaryotes, they form a centromer, they form a chromosome. The copies of chromosomes are unique in number in all vertebrates. They doesn't vary in number. They are constant in each species. So here the eukaryotic chromosome are typically linear, and eukaryotic cell contain multiple distinct chromosomes. The many eukaryotic cell contain two copies of each chromosome, and therefore it's called as diploid. The human chromosomes are also a diploid chromosomes. The DNA supercoiling refers to the process by which DNA twisted to fit inside the cell. Supercoiling may result in the DNA that either underwound or overwound. If it is an underwound, then the less winding of that DNA is happen, and if it is overwounded, then the more than 1 turn per 10 base pairs from its normal relaxed state is observed. The protein involved in this supercoiling, known as topoisomerase, this enzyme helps maintain the structure of supercoiled chromosome, preventing overwinding of the DNA during the cellular process, like DNA replication. The genetic organization of eukaryotes, the following plays the major role in that. These are the chromosome, chromatin, histone, non-histone, euchromatin and centromeres and telomeres. These are the parts of chromosome and they plays the very important role in the packaging of the genetic material of eukaryotes. First is chromosome. The chromosome contains many genes. Genes is a part of DNA, regulatory unit and other nucleotide sequences. Chromosomes also contain DNA bound proteins which serve in packaging of the DNA and control its function. Chromosomes vary in both number and structure. Among the organisms, chromosome is characteristics of every species. Chromosomes are tightly coiled DNA around basic histone proteins, which help in tight packaging of DNA. The next is chromatin. This is a substance within chromosome containing or consisting DNA and protein. The DNA carries the cell's genetic instructions. These are the codes. The major proteins in chromatin are histone, which help in package the DNA in a compact form that fits into the cell or the nucleus. Changes in the chromatin structure associated with DNA replication and gene expression. Histones are highly basic protein found in eukaryotic cells. The nuclei that package and order the DNA into the structural unit called as nucleosomes. They are the chief protein components of chromatin, acting as a spools around which DNA winds and playing the role in gene regulation. So here this is the chromosome. It is formed with the help of this DNA winding and this DNA contains genes. The genes is having functional codes for the different cellular function and formation of proteins, which is compactly wounded around the histone. These are the histones and they are maintain this shape and alpha helical structure of DNA. If we unwound the DNA of eukaryotic cell, it is larger than 2 meters. So that is why the supercoiling of DNA is necessary to fit this 2-meter and more than 2-meter DNA into the nucleus of eukaryotic cells. Non-histones. The non-histones, these are the proteins which helps in a different type of winding process of this DNA. The non-histones are involved in the range of activities including DNA replication and gene expression. They display more diversity. They also differ between different tissues of same organism. The next is centromeres. The centromeres and telomeres, these are the parts of the chromosome. The centromeres are which is found at the center of the chromosome and between the two arms of this chromosome. The centromere responsible for the accurate segregation of replicated chromosome. during the meiosis process. When the chromosome stained, they show dark stained region, that is centomers. The actual location is where the attachment of spindle fibers, telomers. It gives stability to chromosome during the replication. The telomere sequence remain conserved throughout the vertebrates. In human, telomeres contain sequence, that is TTAGGG, repeated around 500 to 5000 times. Certain bacteria also possess telomeres. As its name implies, the telomeres bend around the DNA complementary strand. So this is the genetic organization of eukaryotic cells. Thank you.